Good evening, everyone. My name is Meredith Toohey, and I'm a Nutrition Education Manager at the American Nutrition Association, the ANA. The ANA is very pleased to be hosting Dr. Weston Bussler tonight to explore digestion and the microbiome. Dr. Bussler is part of the research and development team at Standard Process as a nutrition scientist. His work involves translating novel research insights and nutritional product innovations into successful products and educational material for healthcare practitioners to support health and wellness. Dr. Bussler earned his BS in nutrition at the University of Minnesota and continued his education at North Carolina State University, where he earned his PhD in nutrition science with an emphasis in food and agriculture. His research at NCSU focused on the intersection of these three disciplines, where he worked to connect plants to health conditions utilizing statistical genomics. Tonight, Dr. Bussler will lead us on a tour through the digestive system, highlighting features of the upper GI and focusing on the microbiome of the lower GI tract. This discussion highlights highly relevant topics in microbiome studies, including GI barrier permeability, probiotics, and other modifications of the gut microbiome, the GI connection to immunity and brain health, microbial balance, and dysbiosis. The ANA would like to thank our sponsor, Standard Process, for their support in bringing this webinar to you. Visit their website, standardprocess.com, to learn more about their dedication to the field of nutrition and whole food philosophy. Welcome, Dr. Bussler.
Hi, everybody. I'm Dr. Weston Bussler, and today my talk is Start with the Gut, Support the Microbiome, and Digestion. This is a very important subject because for millions and millions of people, digestive concerns are on the rise. So many people actually have a number one of these or have any number of these conditions, including heartburn, acid reflux, GERD, IBS, IBD, indigestion, constipation, diarrhea, abdominal pain, as well as many others. The concerns for individuals can be incredibly complex and all-encompassing for their lives, or they can be relatively minor. They can be just an inconvenience that somebody would rather get rid of. They can be chronic or acute, but all in all, it winds up costing the healthcare system and individuals nearly $136 billion in expenses. It's incredibly costly for us to manage these, and they're really problematic for individuals. Looking at the burden of many of these conditions across the globe, what we see is that in the more developed nations, you have a lot of these associated morbidities. So, particularly in the United States, what we see for irritable bowel syndrome is approximately 20% of the United States adults have this particular condition. It's a really large amount of the population that are dealing with this. Looking at other countries, many of them are also experiencing relatively high proportions of these conditions. While in IBD, it's less frequent on a total perspective, with only about 1.3% of total adults in the U.S. having it, it's also showing up at much greater rates in these more developed countries. So you can see here, Canada and Australia having particular high rates, as well Western Europe. Now, GERD is spaced out a little bit more, but it still does have this pretty large increased prevalence in a lot of these developed nations. So you start thinking about what's common about these nations, what's common about the U.S., what are we doing? It's the GI system. What are we eating? Well, eating is something that we're doing quite a lot of in this. The diet that we have cultivated over years and years, we actually refer to now as the standard American diet, or the SAD diet. And this is characterized by these incredibly energy-dense, nutrient-poor snacks and just highly so there's a lot of the same type of nutrients that are being delivered at really, really high amounts, a lot of the same foods being utilized for everything that people are consuming. It has a high rate of added sugar, as well as very calorically dense saturated fats. But there's a bit of a problem here because these foods, they're hyper convenient, they're incredibly flavorful and delicious for people, and it's really easy to develop some kind of a preference for them. You can have one particular food producer create one standard food that gets shipped all across the world, and it's just the exact same everywhere it gets. It's hyper convenient, but this is the most efficient form of calories that our species has ever seen in its history, and we're just continuously consuming the same things that they're just driving all sorts of problems in our bodies. So you look at how this modern diet influences the GI. Specifically, it has a connection to all kinds of inflammatory conditions and inflammation in general. So this pairs with susceptibility to infection, allergies, as well as autoimmunity. And these certain dietary stimuli have the potential for inducing or mitigating this inflammation in humans. So you have a lot of the problematic foods that are driving it, such as saturated fats, the isolated sugar, excess amounts of red meat. But you then, on the contrary to that, are going to have a lot of things that are conditionally problematic for people. So something like gluten or salt. Some people don't really have the ability to use these effectively. They can wind up causing a lot of problems for individuals. Then certain relative unknowns are specific fats, artificial sweeteners, but this all ties into stimulating that dysbiosis, that gut inflammation, which then has these connections to systemic inflammation, allergy and autoimmunity, infection, susceptibility, as well as many other conditions. Now, contrary to this, you have additional components that can be relatively protective. So certain dietary omega-3s from fish oils, as well as plant-based phytonutrients, have displayed a lot of evidence to help positively control this gut-specific inflammation and be helpful for people. So this is really important because studies show that when individuals have elevations of these inflammatory cytokines that are stimulated by a lot of these negative dietary stimuli, really common in the standard American diet, they have elevations of these from TNF-alpha, IL-1 beta, and IL-6, and these are associated with that poor gut health. So focusing on all the good things that we can give and limiting a lot of the things that, when consumed in excess, can be major issues. It's all about attaining and helping to drive that ideal GI state. That's characterized by effective digestion and absorption of food, a normal and stable intestinal population of microbes, an effective immune status, a functioning GI barrier system, and an overall status of just general well-being. The features and environment of the GI are separated into the upper and lower GI. The upper GI, starting with the mouth and going down from your esophagus, you have the stomach, then the liver, gallbladder, pancreas, all part of this. This area is where you see a lot of that breakdown of food, the mastication of food initially in the mouth, when it gets solubilized by the saliva. As well, you see all this effect on the various macronutrients being broken down, so carbohydrates, proteins, and fats. This is where it all starts. In the stomach, that acidic pH from the HCl, it causes a really low pH that's important for sterilization and many other effects. the enzymes that are produced here, pepsin, amylase, hormones, bile production in the liver, storage in the gallbladder before it's released into the intestines. For early digestion, you have the saliva and salivary enzymes that are really important for many different functions and required for really proper digestion and absorption. Once again, where you see that food initially broken apart, that functional role of saliva includes all sorts of antimicrobial, antifungal, antiviral actions, as well as it's a lubricator. And the buffering, the taste effects, overall digestion of nutrients, particularly amylases, but it's also got some DNAs, RNAs, protease, lipase. But the mucin within the saliva, it really contributes to that coating, and there's also this mineralization effect on various nutrients. It's a lot of important factors, and looking for, there are certain herbal components and food components as well that can wind up supporting this section of the GI. But one of the things to consider is just hydration, simple hydration. So drinking water. Moving down, the acidic environment in the stomach is important, primarily because it begins this process of protein digestion, where the hydrolysis of proteins is occurring. These proteins appear in the GI once they're consumed in food as their normal tertiary complex state. So a lot of these proteins are actually enzymes that perform a specific function. But what happens when you put them in this acidic environment is that tertiary structure begins to unfold. So they unfold and they start to break down to the point where our digestive enzymes are able to cleave the specific amino acids, releasing them for our ability to digest them. This is also part of why the HCl, the acidic environment in the stomach, is so important for the breakdown of foreign microbes. It has a sterilization effect. Some of it can be attributed to that exact protein effect. And this is also going to be very important, this pH here, for increasing the absorption of specific minerals. And when you start to see things get out of whack in this region and the pH changing, you can become susceptible to a lot of microbes. H. pylori, for instance, able to colonize in this location when the pH is not functioning properly. For supporting this gastric activity, it's important to consider things like betanine HCl. That external HCl can really support that overall pH condition. Digestive enzymes that work within a particular pH range are important to think about. Then bitters, so altering or certain bitters, a lot of the plant components that contain these bitter molecules. Vinegar, simply delivering a lot of this low pH. Gentian root, one particular herb that has showcased a lot of positive effects on this gastric acidity, as well as just stress management. Stress is a huge effector for this section of the GI, and it's really important to try to also look at this holistically and target an individual's stress as well with all of these other supportive nutritional aspects. For enzymes, this is where that macronutrient breakdown all occurs. So the carbohydrates, lipids, proteins, this is where you see them be broken down. For carbohydrates, amylases, alpha-galactosidase, beta-galactosidase. For lipids, the lipases. For proteins, you have pepsins, and then peptidase, protease, endoprotease, all necessary for breaking down proteins at various amino acid points. For digestive enzyme support, there's quite a few different types that are helpful. You can just look even at the types of these digestive enzymes once again, but looking at them a little bit further at what exactly they are capable of doing. So certain proteases, they're more of a blunt hammer for trying to break down all kinds of proteins, but some of them might be a little bit more specific. Then you have amylases that can target simple carbohydrates as well as some of the less normally digestible carbohydrates for humans. The sources of many of these, they can come from pancreatin, so just a pancreas derived portion, as well, or pancreas derived supplements. As well, you have the aspergillus, so derived enzymes. So aspergillus is really good at digesting a variety of different nutrients, and so carbohydrates, all kinds of proteins. Those enzymes from this particular mold are capable of breaking down a lot of different things. Then you have the food-derived enzymes, and these are really cool. So you have certain foods, particularly fruits. There's a few fruits that have demonstrated an ability to really break down these proteins. So of them, you have figs containing phycin, papaya containing papain, pineapple containing bromelain. These plant-derived enzymes are really, really efficient protein degraders. There are also many other digestive aids that can be paired with this particular herbal components that have shown to have specific benefits for people. So a few of them, like cumin seed forte, mint, fennel, ginger, a lot of these have very outlined benefits for digestive specific support. Beyond these enzymes, bile is another component of digestion that's very, very important to look at in how it interacts with the upper GI, but as well how it interacts down in the lower GI. So bile is made up of bile salts, lecithin, cholesterol, and bilirubin. It's biosynthesized in a healthy liver and stored in the gallbladder, and we humans secrete about 700 milliliters of it per day. Bile is critical for the digestion and absorption of fats and fat-soluble vitamins in the small intestine. It's used for the removal of cholesterol, xenobiotics, so these drugs and heavy metals and these what you would call toxic components, and or not all toxic components, but these various different components that our body recognizes and tries to get rid of through a number of mechanisms. Various metabolic waste and excess hormones, all of these are removed through, or a lot of these are removed through bile. Then there's also how bile is an important inhibitor for the growth of many different kinds of bacteria. The protection against certain bacteria in the small intestine. Bile is really important for performing that function. So bile synthesis is actually tightly controlled in healthy individuals by a feedback system that assures only the amount of bile needed is made. And for this, microbial interaction also plays a role in that feedback mechanism. But once the bile is released into the GI, most of it winds up actually getting reabsorbed into the ileum. So into the small intestine, it gets absorbed. But a small amount of it will make it to the colon, where some of it's absorbed, and a few bacteria, specifically from Firmicutes, Bacteroides, and Actinobacteria phylums, can actually metabolize it, but the rest winds up being excreted into stool. When an individual doesn't have enough bile circulating, so too little bile, it leads to a lot of malabsorption of dietary fats. So clinically, this can look like fat in the stool. As well, there's greater susceptibility to unwanted bacterial growth. Substances called cholagogues are able to stimulate that bile release. But when there's excess bile, so too much bile that's being expressed by an individual, it may cause things like diarrhea via a number of mechanisms. So altering the water and sodium transport, increasing the lower gastrointestinal motility, damaging the mucosa, inducing mucus secretion, stimulating defecation, all sorts of these negative problems. And for these people, bile binders can be a really big help with this. Excess bile can negatively overall impact that commensal microbiota, because not all of them are able to metabolize it. And as outlined earlier, it is an antimicrobial component. So looking at this graphically, you see this feedback mechanism of normal healthy people, where when you have increased bile absorption happening in the enterocytes, goes back, the feedback loop gets towards the hepatocytes and tells them to downregulate bile acid synthesis. And then the cycle winds up repeating following various fatty stimuli. But during compromised function, what you see is that the bile acid feedback gets disrupted, less bile is absorbed, and this whole system is impaired. So you start seeing increased bile synthesis from the hepatocytes and a lack of absorption in the enterocytes, ultimately moving so much of it towards the colon, where you can now see that increased colonic motility, the visceral sensation, the fluid secretion, mucosal permeability, all leading to these negative effects, a lot of it being characterized by diarrhea. Also, when you look at where would you see this, what sorts of things are causes, there's quite a number of different causes. People who have had some kind of a colon resectioning, radiation injury, gallbladder removal, as well various intestinal conditions. This is something that can be seen with IBS, SIBO, people with celiac disease. And then for others, it's just an unexplained phenomenon at this point, where there's no clear cause, but it's still something that's winding up occurring. We're learning more about it, but what we're seeing from a therapeutic standpoint for a few of these is that from the diet, so this is just looking at diet, the various plants, including high fiber containing plants. So fiber itself performs this function where it can actually bind bile and help remove it. So in these, with a couple of plant components, particularly beetroot and okra fruits, they display this moderate ability to bind bile salts. And this can be supportive in helping to remove the excess bile. But one of the points to consider is in this recirculation process, so much of it winds up reaching the lower GI, but there's been all of these types of effects where it's performing the xenobiotic metabolism, helping to get rid of a lot of negative compounds, and supporting the clearance of these also helps the body just maintain that proper elimination status. For colodogs, jumping back to not having enough bile, in those individuals, some things that you can look at would be like gentian root, gentian lutea, dandelion, ginger, wormwood, herb extract. This is in the Artisma absinthium genus and species, and this is just such an interesting medicinal group of plants in this Artisma genus. It's where you have a lot of malaria medication. It's really, really wild where a lot of these come from, and the effect that you see on the specific bile support is really, really cool. Then you have fennel seed. Some additional things you can look at would be those beets and okra and how those interact. For the lower GI, this environment is characterized by a number of different conditions. The anatomy of it contains the small intestine, the large intestine, as well as the colon and rectum. But looking at it, the features of it, It has a neutral pH to slightly acidic. More enzymes are being utilized and produced, released in this region. The hormones are involved in this location as well. Now you have the selective permeability where we're uptaking nutrients and making sure we keep other things out. So these nutrients are selectively absorbed. But this region also is where you start to see the housing of trillions and trillions of microbes start to occur. So there's microbes throughout the GI, but they really change drastically based on location. And if you look at we'll start in the stomach. You have a really non-diverse and low in number group of microbes. That diversity increases as you go further and further down the GI, with it ultimately reaching the colon, where it's just overwhelmingly so many, many more microbes than the rest of the GI. Furthermore, looking at the layer of the GI, you have the intestinal lumen, and the intestinal lumen is what you would call that external environment within the GI. So this is where all that fluid is moving through and food goes through, where all the external components come through. And it's important to think about your lower GI as an external environment, or the GI tract entirely as an external environment entirely. It's a tube. And this tube, when you consume something, those things aren't fully inside you in the way that if you were to use a needle and stick it into your bloodstream. This is still an external environment. Things have to move through this gut barrier. But in that intestinal lumen, you have all these microbes that live there. And the microbes that live there are going to be a little bit distinctly different from the ones that are housed within the mucus layer, and then ultimately the ones that are within the closest to the epithelial surfaces. So these microbes range within these locations in number and diversity, but also just architecturally in the GI. Looking at that GI barrier a little bit closer, you can see that it's layered with multiple different types of barriers. And these barriers, they need to be able to separate that external environment from the internal one within. And they work together to do this. The first barrier is the microbial barrier. And this is where you have the trillions and trillions of microbes that live within it. So this is where you have all of the good and potentially bad microbes that inhabit the environment. The foods that directly feed the good microbes in here are called prebiotics. A lot of these other microbes are able to scavenge and consume many, many different things. They produce all sorts of compounds that are helpful for us, but then they also can produce some things that we don't necessarily want. So it's a really unique environment within the body where these microbes, they interact with each other. They have their own world, this incredibly violent world where they're teaming up together and crowding out and directly killing a lot of these other bacteria. It's a fight for survival within them. When we classify them, we can classify them based on their taxonomic makeup. So at the highest level of this taxonomy, back when we're thinking about our old high school science classes, you have the phylum level. This is the 10,000 foot view. So it's like, what solar system are they in? Well, for this, it's the different phylums to think about are Actinobacterium, Firmicutes, Bacteroides, Proteobacteria, Fusobacteria, Varroa, Microbia. But you have all these different phylums, and the phylums are going to be something that you can look at as a proportion. So these phylums are made up of many different microbial species, but it's really important we can make general distinctions and look at how these phylums are to just get a general overall picture of the GI and where we're at. As you get further and further down into specificity, you get to the genus and then the species level. The genus and species is where you can be really specific and say, okay, there's these microbes or Bifidobacterium longum. This is one kind, one grouping of bacteria. Then going down the line just like that. But I mean, one example where strain specificity starts to really come into play is something like E. coli. So E. coli is a really common resident microbe in the GI. But the strain specificity is where you start to see how that microbe actually is really functioning. So the strain specificity, you can see really, really big variations in how these microbes are strain to strain. Those can be so much more drastic than what you would see in something like humans, where all humans are going to be the same genus and species, but predominantly they're relatively similar genetically, person to person. You see huge levels of variation across microbial strains. And when we look how this microbiome develops, and the term microbiome actually encompasses all of the genetic material of these trillions of microbes. And the trillions of microbes that make up our GI microbiome, they actually contain far more genetic material than our own human genome. So the layers of complication just continue to add up and add up and add up. But our microbiome is developed from birth, where instantaneously the birthing process starts to really impact how the GI microbial population exists. So you have the birthing process, C-section versus vaginal delivery. It's this initial colonization and exposure. Then you have infant feeding method, where breastfeeding versus formula feeding type, whether or not there's any sorts of supplements being added in. This all is impacting the microbiome development. Then as the life cycle stages change, the various stimuli begin to really impact the microbiome and shift what the needs are. So as a baby, you have a very limited diet, but as you get older, the diet expands and you need to have a more diverse set of functions of microbes to be able to handle all the food or from the microbe, and that's more from the microbial perspective. They are receiving all sorts of different dietary stimuli that they're able to then feed off of, grow, and colonize. Geography is a huge component. So a healthy microbiome can look very different across continents or even regionally, whether or not you live in a city or if you live in the country. Then things like the different pharmaceuticals people are taking on a daily basis. Their diet is this huge component that affects the microbiome. So are you getting enough fiber? Is it mainly plant material? Is there so many refined carbohydrates, high dietary fat? Then you have things like stress, exercise, metabolic stress, physiological or psychological stress. All these can change the microbiome, and many of these are actually modifiable factors. So specifically diet and stress, those are modifiable. But it's not all just bacteria too. A lot of those examples I was giving earlier are just bacteria. You can also think about this fungal biome, and there's even a virus biome, a virome, that's now being started to be characterized. But in the fungal biome, what you see is that there's a lot of different fungi, and they're detectable in almost all sections of the GI tract in about 70% of adults. And there's at least 267 distinct fungal taxa that have been reported in analyses of the human gut. The environmental and commensal fungi are in constant contact with the host, and they provide this myriad of fungal antigens with which we wind up interacting. And during the homeostatic conditions, commensal fungi are kept at the mucosal surfaces and in the skin, while environmental fungi are either killed or they're tolerated when we interact with them. But when these conditions become aberrant, And specifically when mucosal and immune barriers are compromised, the fungi invade these tissues, leading to a lot of dissemination, inflammation, and disease. The fungal dysbiosis in GI conditions is characterized by a lack of the Saccharomyces cerevisiae and an increased proportion of Candida albicans. There's a few controlling factors that are that come into place, both towards the healthy scale and dysbiotic scale for either environmental or commensal fungi. So how our genetics are, the immunosuppression, as well, are you in a hospital? These kind of things play a big role. But then there's also, are you taking antibiotics? Is there a bacterial dysbiosis or any other kind of barrier disruption that's underway? But when we look at them overall, there's a really big desire to classify microbes as either good or bad. And it works in a way, but it's a relatively simplistic classification. So you have microbes that you would call commensals, so these permanent residents and bacteria that provide, or microbes, not just bacteria, that provide a much needed diversity in the GI. Then you have within them a lot of these symbionts, these commensal microbes living in the GI that have evolved to perform beneficial functions. These can include Bifidobacterium and Lactobacilli, as well as F. proud. The Bifidobacterium and Lactobacilli have just been long seen as beneficial species, and they encompass so many of what you would call a lot of our probiotic species. And those probiotics, they're beneficial microbes. They've shown benefits, but they're not these resident ones. So they come in and they're able to influence their effect. But they don't necessarily colonize in the same way. Then, contrary to the symbionts, you have pathobions. Pathobions are these resident microbes that can cause disruptions under normal, or under non-normal certain conditions. Under normal homeostatic conditions, they're actually well balanced with the good microbes. So they're part of that overall microbial diversity. But when something happens and they wind up in a spot they're not supposed to be, so say they get in a microbe that's supposed to be in the lower GI winds up in the upper GI, that can become very problematic for an individual. Or if there's some kind of a depletion in populations of good bacteria and these pathobionts outgrow and they become very, very, they become a higher proportion than they normally would, then they can cause real problematic effects for the host. But beyond that, we have these additional external microbes. These are environmentally acquired ones that are opportunistic colonizers, and they can cause this rapid GI disruption. So thinking of things like Pseudomonas, enterotoxigenic E. coli, Campylobacter jejuni, Salmonella, rotavirus, all of these are able to really cause a lot of big problems. And for microbial balance, a healthy microbiota, we would like to call this homeostasis. So this good balance of microbes, it's a high in both, or high diversity in both species, so the different species of microbes that exist, as well the function of the different microbes. So are they expressing all of the relevant traits that we need? So by having this high diversity in species and function, you have a lot of overlapping effects, and the body's able to get everything it needs from the microbiome, stimulating the immune system the way it needs, training it for whatever relevant thing comes up. These microbes respond to threats and are able to resist those changes under stress. But in a dysbiotic state, you have a couple of different kinds of dysbiosis that can be in existence in an individual. So you can have an expansion of the pathobiome. So those sometimes problematic species can outgrow the others and start to lead to problematic effects. Then you can have a lower level of total species diversity, which can be problematic. As well, you can just be missing a lot of these beneficial microbes or have a very low number of them. These different types of dysbiosis can all occur exclusively, or there can be a little bit of mix between the various different kinds. And how these different conditions, so this dysbiosis, how do these relate to things, to the various stimuli that we have? One of the key effectors of the microbiome is diet. So when you look at diet, what sorts of diets are associated with this dysbiosis? You have in the Western diet that SAD diet. The SAD diet, you see a decreased number of total bacteria. The bacterial diversity is affected directly. Good bacteria, you see bifidobacteria, lactobacilli, those are dramatically reduced in these people that would have a Western-style diet. There's also increase of other species, and then the proportions start to change wildly, and you can see something like a group, a very diverse phylum like the Bacteroides, really start to predominate over the rest. There's many different ways that it can happen, but when you look at it globally, these are a lot of the really big commonalities that you see with a Western-style diet. Moving to what you would look at as a healthy diet, a Mediterranean diet. It has so much great data for how it's protective to all sorts of non-communicable conditions. The Mediterranean diet is high in fiber, antioxidants. It doesn't have too much red meat. There are all kinds of good fats that are being delivered. So in this, you see an increase in total bacteria, increase in the good species, Bifido, Lactobacilli, Roseburia, another group that's really important for particular things that they produce. You can see a really big amount of increased diversity. But then it's not just these two really general diets. Other specific diets can really start to affect the microbiota. One therapeutic diet that people often use, the gluten-free diet, where they're avoiding gluten-containing foods, can actually decrease total bacteria as well, and specifically have some effects on something like the bifidobacterium. And this is where people are doing this for a specific effect, so they can't process that gluten. But there's some unintended effects that could potentially be supported by a particular therapeutic targeting of good bacteria, as long as it's not exacerbating any of the problematic conditions. Additionally, you can look at something like GI function. So this can be very simply looked at through something like the Bristol Stool chart, where you are able to characterize how GI function is occurring based off of how the stool looks, and in associations with these different types of stool, you can see some commonalities across whether it's increased or decreased microbes in individuals that have particular stool. Additionally, condition-specific effects can be really associated to a particular microbiome. And this occurs in conditions that are not only just GI conditions. You're starting to see them occur in cardiovascular ones, where there can be particular microbial profiles associated, as well as liver-specific conditions. Glucose management, so something like where individuals are struggling with overall insulin and glucose response, you can see in association to something like a reduced level of bifidobacterium. In weight management issues, then there's an association that's been outlined as a ratio problem between Bacteroides and Firmicutes. So this is going back to that 1,000-foot view of the phylum, saying that ratio between Bacteroides and Firmicutes is off. It's outweighed far too heavily. And then lower GI specific ones, there's going to be some 1,000-foot view phylum levels that you see change, as well some specific genus species effect are things that you see. So something would be like F prow being reduced. That's a very lower GI specific dysbiosis. For prescriptions that are able to modify the microbiome, you have things like proton pump inhibitors, antibiotics, inhalers, SSRIs, paracetamol, opioids. This study was actually able to outline a lot of the really common pharmaceuticals that are used and how much they can impact the different microbial profiles. So they looked at this huge population and all of the different prescriptions they take and which ones were associated with these microbial changes. And it was really interesting to see that you have certain effects that are being driven by a lot of these prescriptions on the microbiome. Now, one of the ones that's been really outlined to be able to modify the microbial environment, but specifically in a negative way, and it increases that susceptibility to the environmental influences, is antibiotics. Antibiotics can negatively alter the gut microbiota, and that's not surprising when you think about antibiotics. What they do, you're trying to kill bacteria, but it also starts to affect the metabolic byproducts. So they're bacterial microbial postbiotics, and it can start to also have some functional effects, so alter the intestinal integrity as well as the immune response. One particular component that's produced by bacteria called butyrate, it is normally a relatively really a supportive component produced in the lower GI. We'll talk about it in a little bit more detail later, but what you see is that it's particularly impacted in antibiotic exposure. And what that does is it leads to a greater susceptibility, something like a C. diff challenge. So for this butyrate, you see it's really important for the recovery of bacterial overgrowth, the protection of intestinal integrity, and helping to keep that pH low. Looking back at the chemical, or at the different layers of barrier in the GI, one of the ones to remember is that mucosal, that mucus barrier that lines the intestines and lines the enterocytes. This is made up primarily of that mucin compound produced by the goblet cells with this protein core and oligosaccharide side chains with terminal sugars that are fucose and fulic acid that are really important binding sites for a lot of these microbes that latch on the GI. So it's important to remember that this chemical barrier serves a function for helping to protect that GI in times of stress, specifically. Now, moving down the barrier section, we also have the physical barrier. And the physical barrier is made up of all of these different epithelial cells, as well as the goblet and Paneth cells, and they're connected tightly by these tight junction proteins. And if you look here on this diagram, it's actually a relatively complicated mixture where you have all these different components that help stitch these cells together and make that barrier so strong and allow for the selective uptake of nutrients through the cells, and very, very specific things are able to get through only. This last layer of this barrier is that immunological barrier, and the immunological barrier is housed within the GI and is capable of responding to all sorts of threats. So if some microbes start to actually make it their way through the barrier, if there's some sort of changeover or they're just able to get through for one reason or another, then the immune cells should be ready to respond to that area. These immune cells are also really important as stimuli on the outside of the immunological barrier. They're producing things like secretory IgA, IgE, and as well, this is where so many of these different immune cells wind up being recruited and wind up working. So you have all kinds of immune activity that occurs, especially you see it once microbes start to translocate. So furthering on that subject, when we start looking at something like gut permeability, in a healthy GI barrier, it functions by selectively allowing nutrients through and keeping the larger food particles and pathogenic bacteria, yeast, parasites out of the GI. But in a state of hyperpermeability or leaky gut, the tight junctions are loosened, so undigested food particles and pathogens and things can move through and start to activate the immune system, causing inflammation. A lot of these microbes are normally not problematic, but now they're reaching that space where they do cause an immune response being required. And this also can be or be connected to certain individuals' food sensitivities. But certain things like an unhealthy diet, excessive antibiotic use, and stress, they can all impact that intestinal permeability. Furthermore, intestinal hyperpermeability has been associated with a lot of health conditions. Some of the most prominent conditions have been associated with this increased amount of intestinal permeability. Autoimmune conditions like ulcerative colitis, Crohn's disease, systemic sclerosis, type 1 diabetes, liver-related conditions, so primary biliary cirrhosis, chronic liver disease with type 2 diabetes, liver cirrhosis, chronic liver disease, non-alcoholic fatty liver disease, all of them having that really strong association with increased intestinal permeability. Then diabetes, as well, neurological conditions like autism, and digestive conditions like irritable bowel syndrome. This leaky gut hyperpermeability is an expanding area of understanding, and it's really important to consider where it's causing problems and why. So why is this gut permeability potentially such a big problem, and what sorts of things exacerbate it and lead to it? So in the connection to the GI microbiome and permeability, what you see is that this dysbiosis and depleted diversity is directly connected to that intestinal hyperpermeability. So you have a lot of the depleted diversity, lack of symbionts, colonization in problematic species, increased pathobionts, follows up with depleted chemical barrier secretions. There's gaps now in the tight junctions that are allowing a lot of microbes, commensals, and pathobionts to translocate and become a problem. They bypass this barrier, and the immune system has to respond to these constant threats. They perform very, very strong reactions that are really damaging, and if not properly quenched, they can damage the external environment. And if this process continues to perpetuate, it winds up further degrading that GI integrity, allowing more of it, requiring more of a response, and you get this prolonged GI state of chronic inflammation. And it's not only just what's occurring right there, you also are now allowing these microbes, the things they produce, the microbes architecture themselves, to make it into circulation and to start reaching other organs. And this is really important because in recent years, we've outlined this really important connection between the gut and the brain. And this is mediated through the vagal nerve and circulation. The microbiota within the gut and the things they produce are capable of influencing other organs, particularly the brain. So if you think about these microbes coming through, and if they're problematic ones, if they're even killed, if they're microbial byproducts, are reaching circulation, moving through the body, and causing further inflammation. So if it's a bacterial cell wall layer, lipopolysaccharide that stimulates an immune response, if that reaches throughout the body, you can increase the state of inflammation throughout the body. And it's a really evolving area and an intriguing possibility as a therapeutic for all sorts of these CNS and more expansive whole body health by targeting the GI microbiome. It's a really exciting area of research, but it's also a very complicated spot just because these microbes are so complicated. But when we look at how we would support these different microbes, we can start with something like all the different biotics and just learning these terms, the biotics. So starting off, one that most people are all familiar with is probiotics. Probiotics are live microorganisms. They confer a health benefit on the host when they're administered in adequate amounts. Clinical evidence documents this at the various dosage that's being looked at. Prebiotics, the dietary substances that nurture specific changes in the composition and activity of the GI microbiota that favor beneficial bacteria, thus conferring those benefits upon the host. So the prebiotics are very important alongside the probiotics. Then you have things like the symbiotics. So these would be products that contain both pro and prebiotics. Bonus points that they both work together to feed those specific probiotics in the mixture. Then some newer terms, things like phytobiotics. These would be some non-essential plant phytonutrients that are capable of modifying the GI environment. And I like to think about this and try to connect a lot of these more herbal-based remedies as well as the dietary phytonutrients and how those have been used for such a long time and are now being recognized for their very strong ability to impact the microbes in the GI in either a up or a down fashion. So you can target many different ways. Then there are postbiotics. Postbiotics would be the non-viable bacterial products or the metabolic byproducts from a probiotic microorganism or other beneficial microbe that has a biologic activity effect on the host. So these would be a lot of the fermentation products you can think about, and the fermentation product itself has a benefit. Then there are psychobiotics, which are a subclass of probiotics, but these ones confer some kind of cognitive health benefit. And finally, one newer area that's being looked at is what are called parabiotics. Parabiotics are the inactivated microbial cells, which when administered at sufficient amounts, even though they're dead, deliver some kind of a health benefit to patients. Sometimes these are referred to as the ghost probiotics, but they're a really intriguing area of research for the future. Back to probiotics specifically. Probiotics confer a lot of biological effects. They can be done through a number of different ways, and probiotics are going to be important because they compete for nutrients. Specifically, they can compete for prebiotics. They are able to bioconvert all sorts of different metabolites. So certain metabolites, when ingested, might not do anything, but if they get bioconverted into something beneficial, they can provide a health effect to the host. So that would be production of a lot of these postbiotics. Production of growth substrates, so more postbiotics, but these could be very specific vitamins. Different microbes are very important for producing those. Some of them have effects on things like mineral metabolism. They can have direct antagonism effects on other microbes. So they can produce their own antimicrobials called bacteriocins, but they also can do this phenomenon called quorum sensing, where little signals can be sent from microbe to microbe that can convey you're not welcome or some other kind of interaction between the two of them. This quorum sensing is a really intriguing way that these microbes can communicate with one another. Direct competitive exclusion, so you have microbes that can compete for barrier sites, or where they would bind and outcompete one another in a number of different ways. As well, they can influence the function of the GI barrier. They can have direct impacts on the tight junctions or the various other aspects of that GI microbial barrier. They can reduce the amount of inflammation, as well enhance the immunity of the whole body. But one thing to remember about probiotics in comparison with a lot of the species that colonize the GI and are residents there is that most probiotic organisms, when supplemented, they do not colonize the GI tract effectively. So they can be thought of more as tourists. And if you think of the GI as an economy and the residents as permanent residents that live there, the tourists can come in and support that economy. They can support it in a number of different ways, but most likely they're not becoming those permanent residents. So it's important to look at them in that function and how they can be delivering this effect. The probiotic effects that they can deliver, it's really important to consider the strain specificity, because those microbial strains can be so different from one another in their function. A lot of probiotics have varying levels of effects, and these range from the really widespread, where almost every probiotic species does it, to the frequent species level effects, where so this species always seems to do this particular thing. But then you have some of the really rare effects that are very strain specific. And these, in the widespread effects, they can be things like colonization resistance, pH modification, produce really wide-ranging short-chain fatty acids, regulate intestinal transit, competitive exclusion. But then the more rare, the frequent effects would be things like particular vitamin synthesis, bile, salt metabolism, enzymatic activity. They can neutralize damaging compounds. They can reinforce the gut barrier. One of the keys that a lot of probiotic species are known for is their ability to metabolize bile, and that's important for being able to deliver it to the lower GI and have a live microbe once it gets there, as well as particular enzymatic activities. Then the rare effects, so things like neurological function, immunological effects, endocrinological effects, production of specific bioactives as well. So the various bioactives that are really, they can be really key to an effect of a various probiotic species. So for those probiotics, some of the examples are things like Lactobacillus acidophilus. But in particular, you can look even more specific at the strain. So a strain like LA5. In a yeast, something like Saccharomyces cerevisiae has been outlined as a helpful microbe, but the variety Boulardii has been outlined to have particular effects. Same thing with Lactobacillus casei 431 and Bifidobacterium BB12. So there's all kinds of microbes that can be supportive. And when you look at ways that people can get a lot of microbes, that would be a range of beneficial microbes. You can look at some of the more diverse delivery mechanisms with food. The dietary inclusions would be from things like fermented food. But this fermented food has to have live microbes. Otherwise, it's going to be more of any effect would be more of a parabiotic effect or a postbiotic effect. But when you look at the live microbes being delivered in foods, you have all kinds of fermented foods that are available: kimchi, yogurts, and kombuchas. All these things are now starting to get much more recognized for their ability to support health alongside these very targeted, clinically proven strains. And the clinically proven strains, they need to be outlined at the relevant levels to deliver their effects. One of the biotics is prebiotics, and we're going to cover prebiotics in a separate section. I have a full talk that pairs with this one that's dedicated to prebiotics. But one thing to remember is that there are also fermentable things that are fed all the time beyond just prebiotics. And one of the biggest would be fiber, dietary fiber. You have a couple different kinds of fiber. You have more fermentable fiber and less fermentable fiber. It can be classed as either soluble or insoluble. But fiber in itself is actually a very complicated subject. But overall, diets with high amounts of fiber provide a lot of substrates for bacterial fermentation that predominate bacteria that produce short-chain fatty acids and help preserve a functioning mucus layer. And then when you look at diets that devoid of fiber, that mucus that lines the GI is actually a source for microbial consumption. So a lot of the mucin consuming microbes begin to dominate more so than they would necessarily in an individual that has a lot of fiber in their diet. And that can lead to a degradation of the barrier for some of those individuals. But when you look at a particular type of fiber, one that's outlined here is resistant starch. Resistant starch, you can find it in various cereal grains that haven't been overly processed, like oats or buckwheat. As well, you can find it in various other food items, green bananas being one. They're very efficient at driving the production of butyrate. And butyrate is one of these really helpful postbiotics. What we see in diets where individuals have a lot of fiber, in particular have a lot of resistant starch, and a lot of these would be more traditional diets that humans evolved with, is that they actually will see a lot of reduced GI inflammation as well, just overall GI benefits from this. So consuming huge amounts, and we're talking about almost 10x the level of fiber that we currently consume. We're pretty far off from consuming as much as we used to as humans. So for all these different substances, they can modify the microbial populations in different ways. There's a lot of different substances that affect the microbiome. You can have prebiotic ones or not prebiotic ones, as well as some that kind of class both ways. So we were just talking about fiber, but one of the things that you can look at with fiber is you have the less fermentable, which can still impact it, but the more readily fermentable can act almost like a prebiotic. But various prebiotics, many of them coming from this oligosaccharide class of molecules, you can have human milk oligosaccharides, and human milk oligosaccharides will also dive much more in depth in my second talk. As well, you have those phytobiotics, the phenolics and phytochemicals from plants that really impact the different microbes. And on the non-prebiotics, you also have things like probiotics. Probiotics can really affect the microbiome. Proteins and fats, vitamins, specific vitamins can, as well, things like antibiotics really can impact the microbiome. Then we also outlined all those various different conditions, but we're focusing primarily here on the dietary inclusions. So for prebiotics, some really common ones for, you have inulin, galacto-oligosaccharides. For human milk oligosaccharides, the most predominant one is 2FL. And for fiber is going to be in almost all whole foods. Whole plant foods will deliver fibers, but some of the really good delivery mechanisms are going to be things like okra, collards, beets, carrots, oats, certain mushrooms, buckwheat, sweet potatoes. There's all sorts of different places that you can get fiber. Certain ones like psyllium husk and inulin can be isolated and have shown this functional fiber effect. For postbiotics, I keep talking about butyrate, and butyrate is just probably the best example of a postbiotic. So its production benefits the lower GI as well as whole body health. So postbiotics don't just stop at supporting GI. Butyrate is both a marker for intestinal health and acts as that important bioactive. So you can look at, in patients, if they have low butyrate levels, or a proportion of butyrate versus other types of short-chain fatty acids that's far too low, then that can be a sign of some sort of problem. Then what you can also see is that as an important bioactive, proper amounts of butyrate are important because they're a primary energy source for the colon cells. They're actually the preferred energy source by the colon cells. They most efficiently can use butyrate, and that's why it's so important that it's produced locally. When it's produced locally, it can be used as they need. It has a direct anti-inflammatory association. It helps with tight junction maintenance. It regulates colonic luminal pH. So, as outlined earlier, really important following and helping to support defense from various overgrowth of microbes. And it enhances certain mineral absorption. It induces satiety hormone secretion, and it alters gut microbiota. It helps keep a more anaerobic environment. So phytobiotics, the plant phytochemicals that interact with the GI microbiota, these compounds bypass so much of the human digestive mechanisms that allows them to reach the lower GI, where they interact directly with the microbes below. And certain phenolics have shown the ability to drive the growth of microbes that elicit beneficial biological effects, such as Lactobacilli, Bifidobacteria, Akkermansia, and even F. prob. But you can see a secondary effect on the different microbes where these can also have a down-regulating growth effect on problematic species. So you look at how these come into the body. There are these really complex components that are bound within the plant matrix material. They're either difficult to digest, they're bio-inaccessible because of the architecture of the plant, or they're just really large molecules that are not very bioavailable. But certain microbes can break these down and turn them into some more bioavailable nutrients. So you can have the direct effect on the microbes where you're inducing growth and decreasing growth, but then you can also, some of these plant materials, once they affect the microbes, like beyond that, once they're converted, they can be directly impacting human health by way of some of the small metabolites that are produced from these various microbes. So you can have the larger effects on the population or these direct effects. So the various ways that plants we eat can impact the GI. You can first look at that positively shaping the microbiota. Number one, inhibiting the production of deleterious endotoxins, positively modulating the production of those short-chain fatty acids, certain metabolites that are capable of mitigating the harmful effects of foods, so things like oxidants and other pharmacological insults, inhibiting hydrolytic enzymes and pancreatic lipase. This is ways that it can impact the overall GI function. Then you can also have mitigating intestinal permeabilization, so effects on the tight junctions and that barrier function overall, as well the associated transport of various endotoxins that can really initiate the inflammatory response. Then you can also see that these plant materials can modulate the secretion of gut hormones by the enteroendocrine cells. So there's all sorts of different ways and many different plant components that are able to impact the GI. And some of these are microbial mediated. Many of them are. But some of the sources you can look at for various compounds, something like myricetin, grapes, beets, Swiss chard, peabine, various catechins from green tea, certain things like pea vine, buckwheat, and pea vine are going to be sources of quercetin. Buckwheat is a really, really concentrated source of quercetin retinoside. And then certain things like red clover, alfalfa, pea vine, delivering something like, or delivering additional metabolites. These plant metabolites, once again, they can modify the microbial population by both increasing and decreasing it. And when you look at those phytobiotics, I've been starting to incorporate a lot of these more traditional herbal components that have been utilized for their specific effects on the different microbes. So a lot of them have been outlined as antimicrobial to very targeted microbes. And it's really important to look at how a collection of these can have been used historically by our bodies as a way to basically self-regulate this population and control so much of that excess inflammation in the GI and all of the negative effects that that can produce. Thinking about a dysbiotic state and just the person that has just long-standing dysbiosis, there's a thought that you can reset this. You can try to change it. And we know that certain dietary stimuli are able to very quickly and efficiently change the microbiome. You can do it within a week. But when you start trying to do some kind of a long-standing change, what's going to be a really good way clinically to approach that? And this is where a really encompassing program like a weed, seed, and feed program can be utilized to try to reset that GI and get it into a spot where it's prepared to just reset and change. So our GI microbiome, it's a relatively plastic state. It doesn't stay the same. It changes a little bit here. It changes a little bit there. It responds to all sorts of these environmental influences. Diet changes for 2 days. Then the diet changes back for 2 days. It goes one direction, but then it always tries to go back to whatever that baseline state is. And this works both ways, for positive and negative. So in the positive state, you'll actually see that various stressors can really change that microbiome. But your microbiome is always trying to return to normal. And supporting that return to normal is really good when that's a healthy state. But if that state gets too out of whack, that temporary dysbiosis, that can become the primary state. And that becomes the new baseline. And now all of a sudden that new state can resist colonization. But what you see when you're trying to actually change that problematic state, you have to look to something more basic, or not more basic, sorry. You have to look at something that's more long-standing and a more comprehensive approach. So you're trying to pair things like a weeding, a seeding, a feeding for individuals. So utilizing of phytobiotics, things that we know can really regulate some of this growth. Then something like a seeding, or then introduce new microbes that are very helpful microbes into this grouping. So you have things like fermented foods that can be used, as well targeted microbe strains that deliver beneficial effects. Then, alongside this, you would look at supporting the feed step. So having a healthy base diet overall. For particular people, you might have to be doing some kind of a exclusion diet, avoiding their trigger foods, if possible, a diverse fiber diet for that overall diverse GI. Targeted prebiotics like inulin, but then you have things like resistant starch, galacto-oligosaccharides, particular herbals like slippery elm bark But then you also can incorporate a really targeted prebiotic like a human milk oligosaccharide to feed a particular group of microbes. So this is one of the initiatives that you can use for trying to make more of a long-term, trying to support a return to a homeostatic state or a normal biosis from somebody with a dysbiosis. And thank you very much. It's one of the messages I want you to take away from this is that the GI is an incredibly complicated and complex system, and it's really important to think about how the microbiome is interacting with the GI system as well as the whole body to influence our health, and how we can start just utilizing the various different ways that we can target the GI. It's one of the modifiable areas of our lives. We can change what we eat. We can change what we deliver. And it's really important to our whole body health overall.
Thank you, Dr. Bussler. Unfortunately, our time is up. This has been an enlightening evening. Good night and best of health to you all.